Hello, everyone. My name is Lorenza Rimassa. I'm a medical oncologist at Humanitas University and Humanitas Research Hospital in Milan, Italy. It's my great pleasure to introduce this first episode of the new webinar series titled Ongoing Clinical Trials for Biliary Tract Cancer in Europe, organized by the Precision-BTC-Network Working Group 3 with ENSCA, the European Network for the Study of Cholangiocarcinoma, in partnership with the UK CCCR. This is a biannual webinar series that has been designed to provide up-to-date information on clinical trials available for patients with biliary tract cancer across Europe. We will have meetings every 6 months with a panel of experts and researchers. The coordinators of this activity are Angela Lamarca, Anna Saborowski, John Bridgewater, and Jiménez Díaz. And as I said, every six months, we will be reviewing and discussing the most relevant clinical trials in biliary tract cancer that are open to recruitment across Europe, including the eligibility criteria, trial locations, study phases, emerging therapeutic strategies, all the information that can be helpful. The objective of this series is to support informed decision-making for healthcare professionals, but also for the patients and caregivers, by offering clear insights into current and upcoming opportunities for participation in clinical research. The webinars will also serve as a platform to foster collaboration and knowledge exchange within the biliary tract cancer research and clinical community. So it's my, again, great pleasure to introduce our first speaker, that is Angela Lamarca from the University Hospital Fundación Jiménez Díaz in Madrid, Spain, who will present the clinical trials that are now open. And we will have a bit of a discussion after different sections. You can send your question in the chat, and we will be happy to address them during or at the end of Angela's presentation. Angela, the floor is yours.
Thank you very much, Lorenza. I think you can hear me okay. Yeah, okay. So we have prepared a few slides with a summary of the ongoing clinical trials that we have now recruiting across the globe in biliary tract tumors, with a special focus on the trials that are now open, recruiting and looking for patients, and also a little bit of some introductory results of some of the preliminary data that we may have from some of the very recent studies as well. These are my conflict of interest. So as we know, the guidelines for biliary tract tumors have evolved very, very much. These are the most updated guidelines that were updated as an e-update by ESMO last year. And as we now see, we have an improvement in the adjuvant treatment. We also have the incorporation of immunotherapy in the first-line setting for advanced disease. And we have an increasing number of targeted therapies. So all these guidelines are really being changed because of the ongoing clinical trials, and is therefore also our duty to try and really inform the best practice by delivering on these ongoing clinical studies. Going back in the story of adjuvant treatment, let's start for early stage biliary tract cancer. We have the data from the BILCAP clinical trial, which, as you all know, is now our standard of care. This was a randomized phase 3 study that compared capecitabine versus observation in 447 patients and showed a benefit in favor of the use of capecitabine. It's true that there are some pitfalls for this trial, but at the moment, capecitabine is our standard of care. So let's focus first on what is happening in the adjuvant setting. We do not have the results of the ACTICA-1 clinical trial yet, but this is one of the trials that, even though it's not open to recruitment, we really must keep an eye on these results. This one, oops, sorry, let me silence someone who is speaking in the background. I don't know where. Anyway, if someone is finding that person, please, yeah, okay, let me go back. So we have here the ACTICA-1 clinical trial. This one is randomizing capecitabine versus cisplatin and gemcitabine. And as I said, is pending results, but it will be really important to really keep an eye on that trial, because it could really be practice changing, even though recruitment is now completed. The other very important clinical trial in the setting of the adjuvant setting is the ARTEMIDE clinical trial. ARTEMIDE Biliary 01 is a clinical trial that has just completed recruitment as well in last week. No more patients are now being recruited into the study, but this was a randomized phase 3 clinical trial that was comparing capecitabine with placebo versus capecitabine with rilvegostomig. So let me show you give me one sec, because I'm first going to try. Someone is speaking in the background. I don't know where.
Yeah. Rob Goldstein. Please unmute him. Thank you.
I cannot. Anyway, if someone can please stop his microphone, that would be great. If we cannot, I will continue. Anyway, so the ARTEMIDE clinical trial, randomized phase 3. After ACTICA is going to be the most important clinical trial in the adjuvant setting. And as we can see here, what we are testing here is immunotherapy in the adjuvant setting. We have rilvegostomig, which is a bispecific antibody targeting both PD-1 and TIGIT, and is placebo-controlled. All patients are getting the capecitabine, but we will have the answer to whether immunotherapy in the form of rilvegostomig is positive or not. This trial was recruiting patients with cholangiocarcinoma and gallbladder cancer. And as we can see here, the main inclusion and exclusion criteria are quite broad. We are very similar to BILCAP, including patients with NET and NEN stage, as far as this was an R0 or an R1 resection. And the primary endpoints includes recurrence-free survival and overall survival. And as I said, that trial, we still do not have the results, but the recruitment is now completed. As of last week, no more patients are allowed. So a very, very fast recruitment into that adjuvant trial. Let's now move into the systemic therapy, into the advanced disease. In here, we have a lot of changes over the last year. We have, as I said, the incorporation of immunotherapy. We also have targeted therapies, and I will mention as well some trial in local regional strategies as well. Local regional strategies, as you know, this is especially mostly for intrahepatic cholangiocarcinoma with liver predominant disease. We had some very promising data from a phase 2 study in the past that was combining SERT with cisplatin and gemcitabine. These are some data from Dr. Edelin, showing very good or attractive data with a combination of the radioembolization and chemotherapy. Despite this promising data, this was a small phase 2 study, not really changing practice very much, and therefore local regional therapies are still not very widely utilized, but we do have ongoing clinical trials in this setting. And we have, for example, the IMUY trial, that is a phase 2 clinical trial. Again, there is no comparator arm. Is a trial with 2 different cohorts where radioembolization is combined with durvalumab in arm A or durvalumab plus tremelimumab in arm B. This trial is very, very advanced in the recruitment, and I believe only a few patients are awaited for completing recruitment. But it's good to see that we are also delivering on trials for liver radioembolization and local regional therapies. And now, just let's go into the systemic therapies. There are a few things ongoing in the area of systemic therapies for the advanced disease. One of the trials that has already reported some of the preliminary results that I wanted to highlight is the Tourmaline study. This trial is important because of various reasons. One is the fact that it's providing us data for the use of durvalumab with other chemotherapies that are not cisplatin and gemcitabine. As you know, the approval of durvalumab was based on the TOPAZ clinical trial that showed benefit of adding durvalumab to cisplatin and gemcitabine chemotherapy for biliary tract tumors, but we didn't really have much data on which is the activity or the potential benefit of durvalumab when we are using other chemotherapy. So Tourmaline was having some patients in that scenario, gemcitabine or any other chemotherapy. As we can see here, there is a variety of different chemotherapy schedules that could be allowed. Gemcitabine monotherapy. There was also gemcitabine with oxaliplatin, gemcitabine with carboplatin, gemcitabine with S1, or even gemcitabine, cisplatin, and albumin-bound paclitaxel. There was also one specific cohort for patients with performance status 2 who were receiving cisplatin and gemcitabine. Again, this was a patient population that was not included into the TOPAZ trial because only performance status 0 and 1 patients were recruited. So again, in Tourmaline, we have this PS2 cohort. And finally, we also have data for ampullary tumors because ampullary were excluded from TOPAZ. They are now included in Tourmaline, and all these different cohorts are really being tested with a combination of durvalumab. As I mentioned, some preliminary results have already been reported. Here we have some data from ESMO GI 2025. As we see, majority of patients were performance status 0, 0 or 1. There is a big proportion of patients from Asia, almost 75%. And here a variability on the different types of chemotherapy that was utilized. I think it's important to see here that the response rate is quite consistent regardless of what chemotherapy is being utilized. It's true that the non-platinum chemotherapy is the one that has the less of the response rate as expected. This will be the patients receiving gemcitabine alone. And as we know, the role of gemcitabine alone is probably more limited in the biliary tract right now, but any other combination with any other platinum or even triple are really getting very similar response rates. And more importantly, the toxicity was also assessed. And in this patient population, where we are expecting a different comorbidity profile and maybe more risk of toxicity because of those comorbidities, and that being the reason why they are actually not cisplatin eligible, what we see in this table is the summary of the adverse events. And as we can see, the adverse events are very similar to what we have already been reported. There is not really an increase in the profile or a change in the profile of the adverse events that we see, regardless of the chemotherapy backbone. The other important development that is happening in the field of systemic therapy. First, we have already mentioned the combination of durvalumab with other chemotherapy strategies. Now we will go into alternatives to durvalumab. So we have, for example, the development of the Gemini clinical trial. The Gemini clinical trial is now open to recruitment and very soon, hopefully opening again a slot for the biliary tract cohort. This study had also a parallel cohort in hepatocellular carcinoma, and it's a study where 2 bispecific antibodies are being developed. So the first bispecific antibody is volrustomig. This one is a bispecific PD-1 CTLA-4, and the second one is rilvegostomig, that, as we mentioned previously, this is the one from the adjuvant study as well, is anti-PD-1 and anti-TIGIT. In the biliary tract cohort, there was cohort 1 testing the rilvegostomig together with gemcitabine and cisplatin, and cohort 2 testing the combination of volrustomig, gemcitabine, and cisplatin. This first part of the study has completed recruitment, but it's very expected that very soon new slots will come up for the biliary tract for further development. At the moment, the HCC part is almost completed as well. There were some preliminary results presented with the rilvegostomig in biliary tract cancer, a total of 30 patients that we see from one of the cohorts that I was mentioning in the previous slide. And in general, very expected or kind of expected baseline characteristics, nothing really surprising here. And look at the waterfall plot. What we have is a consistent response rate with a 31% of response rate, very similar to the TOPAZ data, with a median progression-free survival of 8.3 months. It's still quite very early, really, to see the activity, but I think looking at the spider plot, spaghetti plot, what we also see is the fact that we have responses or reduction in size that even if they don't reach partial response, they are quite maintained on time. So again, looking into the immunotherapy, we see responses that are really there for quite a long period of time. The toxicity profile, again, very good toxicity profile, nothing really that will come up from the cohort of rilvegostomig with cisplatin and gemcitabine. And we will need to await for more patients, because it's true that 30 patients is quite a small sample size here. One of the very, very hot topics is the HER2, the development of anti-HER2 for biliary tract tumors with HER2 amplification. As you know, these are mainly extrahepatic cholangiocarcinoma and gallbladder cancer. And we have two different compounds that we should really know, and that we really must bear in mind, because we have two ongoing clinical trials being developed in the setting and recruiting patients across the globe. First, we have the zanidatamab. This compound is now approved in the second-line setting, monotherapy for patients with a HER2-amplified biliary tract cancer. And as we see, is an antibody against two different domains of HER2, bispecific antibody anti-HER2. The second compound that we have has a different mechanism of action, is the trastuzumab deruxtecan, is an ADC, anti-HER2. And again, this is also approved as monotherapy for a previously treated advanced biliary tract cancer. And as I mentioned, each one of these compounds is now being tested in the first-line setting in a randomized clinical trial. So let's focus first on the zanidatamab study design. Here, what we have is first-line advanced biliary tract cancer, HER2 amplified, where patients are randomized to receive cisplatin, gemcitabine, and PD-1 or PD-L1 inhibitor, plus or minus zanidatamab. This clinical trial will continue recruitment and is now recruiting patients. So really important to look into whether this trial is open at your site or any other closer sites, because we really must recruit into the HER2 studies. There is a second ongoing trial recruiting in parallel, so we should really find patients for both and make our best effort to try and answer these two different questions. This second one is with trastuzumab deruxtecan. So as I mentioned previously, different mechanism of action. And please look into the study design, because this one is slightly different. It is a first-line study, very similar to the zanidatamab, patients with HER2 amplified biliary tract cancer who are randomized to receive. There is one arm that is receiving the trastuzumab deruxtecan with the rilvegostomig. Here, we are not using durvalumab or pembrolizumab. The immunotherapy that is being tested is the rilvegostomig, which is, again, the bispecific antibody that I mentioned in the prior two clinical trials, in Gemini and also in the adjuvant Artemide study. The second arm that we have is the trastuzumab deruxtecan alone, without immunotherapy. And the third arm is the combination of cisplatin gemcitabine with durvalumab, which is considered the control arm in this study. In both studies, as we can see, the control arm is cisplatin gemcitabine, here with durvalumab, and here with durva or pembro, investigator choice. Both studies recruiting, and really important to identify whether they are open at your site or any closed sites, because this is a very good opportunity for our patients. Another one of the very important targets that has been identified for biliary tract tumors, especially intrahepatic cholangiocarcinoma, is the IDH1. And for this, we have the data from the ivosidenib that, as you know, is approved and funded in many countries for IDH1 mutant cholangiocarcinoma that has already progressed to prior therapy. And what we saw in the Clarity clinical trial was an improvement in progression-free survival, even though it's true that the response rate was quite low, less than 5%. Sorry. Let's see if it works. Okay, I hope it goes to the next slide soon. What we see in this, at some point it will work. So what we have seen with the development of the ivosidenib here, there have been two different clinical trials being performed at the moment that are quite relevant. The first clinical trial that I wanted to mention is the PROVIDE phase 3 clinical trial. This one is very similar to the prior Clarity study in the setting where this is developed. We are again in the second line or third line setting after progression to, let me there you go. Okay, here. So the PROVIDE clinical trial is a phase 3, second or third line setting after progression to prior chemotherapy, patients with an IDH1 mutation that are receiving ivosidenib. And as you see here, this was a phase 3B clinical trial. It was really aiming to confirm the activity that we had seen in the CLARITY study. It's not a randomized study. This is the baseline characteristics, very similar to what we expected, and this is the progression-free survival and the median overall survival. We have a median PFS of 4.7 months, presented very recently, just before summer in ESMO GI, with a median overall survival of 15.5 months. So kind of confirming the activity that we were expecting. Again, partial responses are low, 5.7%, also in the PROVIDE clinical trial, but in general, an important study confirming the activity of the ivosidenib. And again, in this topic of the IDH1 mutant, this low response rate has been one of the important criticisms that we have had for ivosidenib for quite a long time. So the unanswered question was whether we should combine the ivosidenib with chemotherapy or with immunotherapy or some other alternatives. And based on this rationale is the design of this ongoing clinical trial that is now again open to recruitment in many countries and in many sites across the globe. Again, there was a safety assessment with 6 patients that is now completed, and now the phase 2 expansion has been opened. We actually had the site initiation visit 1 hour ago at my site. This clinical trial is in the first line setting, combination of ivosidenib, cisplatin, gemcitabine, and durvalumab. Is looking into the safety and the activity of this quadruplet treatment in the first line setting. Important topics here is the fact that now we should really identify not only the HER2 amplification, which may be a little bit more straightforward with immunohistochemistry, but also an IDH1 mutation before we start treatment. This clinical trial is allowing one first cycle of cisplatin, gemcitabine, and durvalumab to be given without the ivosidenib, and including the ivosidenib before cycle 2. This way, not really delaying very much the initiation of systemic therapy and allowing a little bit of time for the identification of the IDH1 mutation. But definitely a very important clinical trial to bear in mind. And again, I encourage you to identify where this one is open, close to your site, to really refer patients there. The other very, very important target is the FGFR inhibitors, and we have heard a lot about different FGFR inhibitors. As you know, we have pemigatinib and futibatinib approved, and I wanted to mention this ongoing clinical trial with tinengotinib. Tinengotinib has shown to have a different resistant profile or a different mechanism of action, let's say, and therefore is active even in the presence of some of the resistance mutations where maybe other FGFR inhibitors like pemigatinib, infigratinib, or futibatinib are not active. And with this background in mind, there was testing of tinengotinib for patients who had received a prior FGFR inhibitor. And we have some of the activity data here. We have here the activity of patients who had acquired resistance after prior FGFR inhibitor. Is not a large number of patients, but we have 10 patients and 4 of them achieve a partial response. So it's quite a significant reduction in the size of the tumor, a significant partial response rate for patients who have already received a prior FGFR inhibitor. And with this in mind, the phase 3 clinical trial was designed, is now open and recruiting patients. In this setting, what we are doing is going into the post-FGFR inhibitor scenario. Patients who have already progressed to pemigatinib or futibatinib are randomized to receive tinengotinib or a standard of care chemotherapy, which will be FOLFOX or FOLFIRI. It's a randomized study. There was first a setting of testing 2 different doses, and now the 10 milligram dose is going further into this randomized phase 3 study comparing another FGFR inhibitor after a prior progression to FGFR inhibitor versus chemotherapy in that setting. And all these or in wider spectrum of molecular testing and targeted therapies is also being explored in the SAPHIR clinical trial, which is not really looking into first line or second line, but more into in the middle, in the maintenance strategy. So in this clinical trial that is being run in 3 countries in Europe, including the UK, France, and Belgium, what is being done is patients are started on cisplatin gemcitabine with durvalumab, and during the first 4 cycles, NGS is performed, and then after cycle 4, patients are randomized to continue the standard of care or to go into a targeted therapy in the maintenance setting. The aim is to recruit a total of 78 patients randomized, and for that, the expectation is to screen around 400 patients. So a very big effort of screening that I believe is going very, very rapidly. And as we see here, a very targeted therapies that are being explored in these clinical trials in this maintenance setting as well. There are other targets, and I will very briefly go through some trials that are ongoing and looking also for patients with biliary tract cancer, or that really we should do our best to recruit biliary tract tumors patients into these clinical trials. KRAS G12D, for KRAS G12D inhibitors, or also for pan-KRAS inhibitors, are a very important target for biliary. MTAP loss has also been identified in GI malignancies, including biliary tract tumors, and we see here that some of the preliminary data with PRMT5 inhibitors in the presence of MTAP loss were actually patients diagnosed with cholangiocarcinoma. Some of the PRMT5 inhibitor trials in the phase 1 setting especially are now recruiting patients, so please bear this in mind, and if you have a patient with an MTAP loss, try and recruit those patients in those trials. There are many other trials, especially with ADCs, that are looking for patients in phase 1 setting, but also specific for biliary tract tumors as well. And with that, you know, there are some very promising targets: HER2, Nectin-4, Claudin, Troponin 2, and c-MET are some of the most important targets for all these ADCs that are now being developed. And this is my last slide. I just really wanted everyone to be encouraged to join the ENSCA effort for the development of new advances for cholangiocarcinoma and other biliary tract tumors as well, and to join our cost action. As you know, we are here today representing working group 3, but there is a huge amount of work being done by all the other working groups, and really I think everyone has some space in this cost action if you have an interest in biliary tract tumors. And that's all from me, Lorenza. Very happy to go into discussion.
Thank you so much, Angela, for this presentation. It's great to see how many trials are ongoing and actually how fast the recruitment is going in this trial. So this is a clear evidence that there is a need of a new treatment. I would like just to remind the audience that this presentation is being recorded, so you will have the possibility to re-watch the presentation in the Precision-BTC-Network website. There is a question in the chat, and then maybe we can go through the different trials. Davide is thanking you and is asking if there are trials with the tyrosine kinase inhibitors as second-line therapy, or trials involving NK cells, immunotherapy with NK cells.
Yeah. So as far as I know, if those trials are ongoing, they are not specific for biliary tract tumors. I may be mistaken. For TKIs, I don't really think there has been a lot of developed, unless there is one specific rationale with one specific target. But not there were a few clinical trials in the past. We had, for example, the GSPF with a c-MET inhibitor, and that was not really promising. In the setting of a non-biomarker-driven scenario. But I don't think there are any TKIs right now for biliary tract. We had also the regorafenib results that were not really practice-changing, and I don't think we are pursuing that strategy. Regarding immunotherapy, a lot of things are happening. We have here, for example, open a clinical trial with cell therapy for patients who have an expression of PRAME. And again, this may be something not only for biliary tract tract tumors, but in the setting of cancer that is really evolving very, very fast. I'm not specifically aware of any clinical trials with NK cells, but well, I'm sure that there are some in solid tumors. And I think the very important message here is that we should really try and recruit patients diagnosed with biliary tract tumors in all these very early phase clinical trials. So we really have the data on the activity.
Yeah, I agree. So Vincenzo would like to make a question verbally. Can anyone open Vincenzo Cardinale's mic?
Yeah, my audio
okay,
is fine. Okay. Thank you very much, Angela. Congratulations. This was very great overview and very useful for, I think, all of us, really. Thank you so much. And also, this initiative is great. Congratulations to the organizer, to Cost Action, ESCA, really. Angela, I have many questions, but I will just do one that is probably very broad. But the question here is that, cholangiocarcinoma is very much different with respect to many other cancer. So I will be focused on cholangiocarcinoma and not on biliary tract cancer. So especially when we consider basket trial, no? Because probably cholangiocarcinoma and BTC are the most heterogeneous that you can include in a basket trial. So the idea is this one. So when we consider combined therapy and you have checkpoint inhibitor plus anti-VEGF monoclonal antibody plus GenChis, or as you show nicely, now we have also EDH1. Okay. So my concern is that if we don't select the patient based on some biomarker, we will increase enormously the side effect and reduce the safety, and we didn't gain anything about effectiveness, because what we'll learn from the biology, pathobiology, is that this target, it's not, are not isolate kind of. You can expect that EDH1 mutated cancer is intrahepatic, is hot, there is a lot of mutation, surely will respond very good to checkpoint inhibitor. While on the other hand, you may expect that in extrahepatic, in some intrahepatic like large cholangio, you will not have any kind of response. So the main question is this: we have this issue, and this is pathobiological and clinical, and if we don't have any possibility to trace the response also based on biomarkers in the tissue, we will never get there when we say this is the right patient or this is not the patient because we're not safe for this kind of therapy. This is my concern. I want to discuss with you and the others about this.
And your question is mainly around immunotherapy, right, Vincenzo? It's around checkpoint inhibitor.
Exactly, because you know very well, we have this nice review together that we know that it's very different, the expression of biomarker for immune checkpoint inhibitor in intrahepatic versus peripheral and distal.
Yeah.
And we cannot consider it small versus the large. And thank you for asking, but we should advocate for this. Okay, thank you.
I agree, because we have established the combination of chemotherapy with checkpoint inhibition as a standard of care for all patients, but we don't really have a biomarker to identify who may benefit most from this strategy. And the truth is that we have some patients who do benefit and some patients who really get into very high number of maintenance immunotherapy, but there are also patients who progress very rapidly and we don't really know who they are. And what we are now doing is incorporating targeted therapies into this triple combination, even if we don't, even before understanding really how the checkpoint inhibition is working, we are now incorporating targeted therapies like the ivosidenib trial that I was mentioning. So in a way, yes, I agree. I would like to have a biomarker, and I would like to have the use of checkpoint inhibitors a little bit more driven by a biomarker. But all the efforts that have been done have failed. And the truth is that we don't have a biomarker. And the truth is that this is also happening in other cancers as well. Not all the cancers have a biomarker for checkpoint inhibition. To me, one of the important questions is also what to do with the checkpoint inhibition at progression. Shall we continue when we change the backbone chemotherapy? What do we do when we, in the setting of an FGFR inhibitor? Are we going to do second line with FGFR inhibitor alone? Should we continue the checkpoint inhibition? I think there are so many questions, not only around the biomarker, but also about what to do in subsequent lines of therapy with the checkpoint inhibition. And without biomarkers, it's very difficult that we answer those questions.
Thank you, Angela. I just want to say just one thing. Thanks to ASCAT and cost action, we may have, because if you see the paper by Guido Carpino in histology, for example, past positivity and the large bile cholangio are very, very very progressing, very different kind of tumor. And we may gain a lot of information. So we should really advocate for have information and put all the effort to raise the good amount of sample histology to have this biomarker.
I agree.
Thank
you. I agree.
So waiting for other questions. So I have a couple of questions going back to the adjuvant setting. So we are waiting for the results of 2 trials, one with a cisgem and then the artemide with capecitabine plus or minus rilvegostomig. First, 2 questions. First, do you have an opinion on if more aggressive chemotherapy can be helpful in the adjuvant setting versus immunotherapy, considering the immune microenvironment and so on? And the other question is, if one of these trials will be positive, either one or the other, how this will be changing the approach in the advance, because I don't think patients will be cured, so eventually they will relapse. And then
I hope I had the answer to those questions. So first, I think we already have results that were not practice changing from a more intense chemotherapy. And we had, for example, the GEMOX chemotherapy versus observation, and that was a negative study that did not show any benefit from the gemcitabine and oxaliplatin in the adjuvant setting. With that in mind, I think it is a real feeling that Actica could be negative, but who knows, because cisgem and GEMOX is not necessarily the same. Actica has a larger number of patients, so probably more of a statistical power as well to identify differences. So I think we really will have to wait. But what will we do if patients receive cisgem in the adjuvant setting? What are we going to do in the first line setting? And it's very difficult. And I think we will be in a very similar scenario to where we are now in pancreatic cancer, where FOLFIRINOX is the standard in the adjuvant and is also standard in the metastatic setting. And depending on the time to recurrence, we may re-challenge or we may go for straight into a second-line FOLFOX chemotherapy for biliary. So I think we will really have to await the Actica results for that. And regarding immunotherapy with the artemide trial, again, the artemide will bring immunotherapy into the adjuvant setting. And what do we do in the first-line setting if we have, in the metastatic setting, if we already progressed or relapsed despite immunotherapy? And it's going to be a very similar scenario where I think looking into the time to recurrence will be probably key and will give us the answer. But my question is even more challenging. What if both are positive? And we have Actica positive, cisgem, and then we have the capecitabine with the immunotherapy. And what are we going to do? Because I don't think anyone was expecting artemide to complete recruitment before we knew Actica results. Yeah. And that has actually happened. So who knows?
It's really difficult. We will be in the same situation as in other cancer types when we have different combinations, active, but not compared.
Compared one to another.
Yeah, one to the other. So really.
Yeah. Maybe if I may ask a question, a bit of a bird's eye view, Angela, thank you so much. And I know there's also Lorenzo and John and Juan, Vale, you all have so much experience with designing trials and getting funding for trials. And I always feel that we have so many ideas. Some of them may be good, But it's always a stretch between we may propose small trials, which would be feasible, but they might be less meaningful. And what we would rather want are the larger, more randomized trials, but they are incredibly expensive. So do you have any insights, any tips to share how to approach this dilemma, maybe also how to speak to industry and pitch your suggestions and try ideas?
John, do you want to take that one? The voice of experience.
Well, we've always gone for bigger is better. Okay, we've always gone for the single big questions in all of these scenarios, and yes, it's cost a fortune and it's taken us a long time. But having said that, we are still established for the most part the standard of care through those studies. So it's only through those big studies that you can do it. And I think pharma can be persuaded to help you with these studies. They can. Yeah. You just got to just keep at it. Just have the faith. Have the faith and believe that you can do these big studies, which we can. I mean, the advanced disease studies, the Artemide recruited ahead of time. The adjuvant studies all recruited ahead of time. Biliary tract cancer is not rare. Okay, everybody out there says it's not rare. And in our centers, with the increasing specialist expertise that we're developing, the people on this call know more about this cancer than other people. We are de facto generating centers of expertise across the world, which can run these big studies very easily. I'd go for the big study every time.
I agree. I think Juan wants to jump in.
Yeah, thanks, and excellent presentation. And just to really add on to John's answer, and specifically, it really depends on two things. Obviously, there's the academic question, which we're all interested in. Can we improve the outcome of patients, reduce the relapse rate, and improve overall survival? But to answer the second part of your question around how you get a new agent, it really depends on where the drug is in their life cycle, because the pharmaceutical companies will also be thinking, is this going to lead to a new license indication? And if they are able to see that there is a line to approval, they may want to run the study themselves. If, however, we're asking a question around a drug that may be, much older, but is academically interesting, or there's some good rationale, where there probably isn't interest from the pharmaceutical company, but there is from clinicians, then as an academic study, I think we've now moved from a decade-long study like BILCAP to a very fast recruiting study like Artemide. So I think the key there is collaboration. And going now beyond the UK, these are now international studies, and I think within Europe, you'll be able to open enough centers to recruit to those clinical trials. So I think the feasibility has now been demonstrated. You probably don't have to have that big discussion with pharma to try and argue that this is possible to do. I think as a community now, we've been able to really show that we can recruit into these studies.
Thank you.
Okay, we don't have another question. So I have another question on the combination of local regional and systemic, because we have several small trials in that setting. Let's say promising results, but nothing really convincing, at least in my opinion. Now there is the trial with the SIRT and the selective intra-arterial radiotherapy. So do you think maybe SIRT or TARE can be different compared to TACE, because the prior trials were mostly TACE?
I think local regional therapies are a very good example from what Anna was asking. Are we going for trials that are feasible to recruit, a small sample size, but not really informing on practice? When we really know that we should go for a big randomized trial comparing the standard of care with the addition of radioembolization. And I think local regional is a very good example of that. And we have had multiple discussions proposing academic studies, pharma-funded studies in the setting of local regional, going with a good clinical trial design, but a big number of patients that we know will need a big budget because of the patient population that we are targeting. To my eyes, what we need for local regional before we call them a standard of care is a properly designed randomized clinical trial. Even if it takes some time to recruit and some money to get the trial done, that is how we are going to get the answer. Otherwise, we will never really be able to get all these treatments incorporated into our guidelines, and most importantly, properly funded for all patients to have access to it. Because to my eyes, having a small trial that may allow some patients accessing that treatment in some centers is not really changing the practice across the globe, and we should really pursue that. So I think that's the perfect scenario to Anna's question as well.
Yeah. And I agree, because, for instance, there are some data about CERT or TER, but for instance, in Italy, but maybe in other countries, it's not reimbursed. So we cannot use for patients with intrahepatic cholangio, while we can use for instance, for HCC. So yes, I agree.
Yeah. And I use this opportunity, Lorenza, because Bas, who is one of the members of ENSCA and COST, and there is also a recorded webinar, if you want to watch it, is actually putting together a grant proposal for a European grant for answering questions on local regional therapy, in these cases with the intraarterial pump chemotherapy for liver-directed therapy combined with systemic therapy versus systemic therapy, including a randomized trial in cholangiocarcinoma. It's a very big effort. It will need a big budget, but again, it's done within the ENSCA network and this cost action network as well. And I encourage anyone who may be listening, who is interested in the topic, to go back to the prior webinar in that topic and have a look, because it's, again, this set of mind of local regional therapy, but we need a randomized clinical trial. So let's go and do our best to try and design it and get it done.
So I go on with Vincenzo, another question.
Thank you, Lorenza. So the question is also a comment based on this very, very, very interesting discussion about clinical trials, because Professor Varley nicely highlighted that we may consider an alternative approach based on rational and basic study to run academic clinical trial when new things will come up. And I think we have a great opportunity indeed in our setting, that is to implement the actual World Health Organization classification, because for sure, if we continue to move forward with new demonstration based on our collaborative effort that is unique, we can say, all over the world, then we may do exactly what Professor Varley was envisioning. Okay, if we finally have the evidence of the clinical relevance of this classification, we may think new clinical trial in academic setting. Because, you know, Angela, I agree that the big is the best, but I think we should have a balance between these two different kind of approach that I think should be synergic somehow. So this is my comment. I don't know.
Yeah, no, definitely, Vincenzo, but we cannot forget and stay on the small clinical trials all the time. So I fully agree. You may have a small clinical trial looking into biomarkers or some specific questions. Even building translational research into all these randomized trials is very, very important. But we should not stay comfortable with having, I don't know how many phase 2 non-randomized may we have in local regional. Too many, and none of them has really changed practice. So I agree, we need a little bit of a balance. It's not only randomized trials.
Angela, but I want to highlight that we should provide the rationale to do what Professor Varley has envisioned. We should provide a rationale. We are going the same good direction. And indeed, it's nice to have a multidisciplinary network as we are. For example, my point of view is the one of gastroenterology, but there are great basic scientists in our so we are going exactly in that direction. But we should advocate for this. The foundation, the ESCA, IMMF should advocate for this, because otherwise, I think we will rely too much on the industry. Okay. This is my personal opinion, but I see Professor Vallée also was same. Thank you.
I agree. Actually, one of the things we have commented on many times is the fact that the adjuvant trial is using a compound that we don't really know for sure if it is active in biliary tract tumors. So yes, there may be a rationale for incorporating immunotherapy, of course, but is rilvegostomig the right compound, or is just the compound that may make sense from an industry point of view? Totally agree. I think we're running out of time, Lorenza.
We have 5 minutes. Last question.
Okay.
One question. Systemic therapy, advanced disease, patients with molecular alterations. Would you go for all in first line, like the ongoing phase 3 and also phase 1b trials with ivosidenib and anti-HER2, or a sequence like the Saphir ABC10?
Anna, do you want to take that one or me? Oh, John wants.
We're only doing it sequentially in ABC10 because of the ongoing problem of getting your result back in time. That is the primary reason we're doing it in that way in ABC10. I think the data, in fact, from that you presented, Angela, from Clarity and then Provide suggests that actually we're getting a bit better at finding these targets and using them somewhat sooner, and the outcomes are, if anything, slightly better. I mean, I feel very strongly that ABC10 is likely to be positive, and the idea of bringing these targeted therapies closer up front with all the added extras, like, you know, getting the surgery, getting to other, you know, better endpoints. I think bringing it up front is the way to go. Practically very difficult still because of the testing.
Yeah, I think the testing really is going to be very challenging for bringing them to first line. Today, when I had the SIV for the ivosidenib trial in the first line setting, my next call was to the pathology department, because now that the trial is open, we really have to have a clear strategy of how are we going to, you know, test these patients as soon as possible, as quickly as possible, because I only have 3 weeks from starting CISGEM to really get in the IDH1 mutant patients into the ivosidenib with the CISGEM and the durvalumab. So it's going to be challenging. And my question also, or my concern, is then, so we start CISGEM durva with ivosidenib or with whatever in the maintenance, and when they progress, then we also have FOLFOX, only have FOLFOX or FOLFIRI. You know, we are utilizing so many of our tools at the very beginning that sometimes feels a little bit scary of what are we going to have left.
If I may comment on target, but also bring it in first line, I think this is definitely the trend, right? There you showed several trials that are using this strategy, and I think it's good thinking, forward thinking. And I think there's one thing that we always have to keep in mind, is that it's such a genetically heterogeneous disease, and we already understand that extrahepatic is not intrahepatic, it's not gallbladder cancer. I think they have different biologies. I think they are different cancers, and even within the entities, they are quite different from their tumor biology and aggressiveness, for instance, also depending on the genomic constellation of the individual tumors. And what we are still lacking is really good either retrospective or even prospective, that would be wonderful data, that tell us like what is the clinical outcome for patients with FGFR fusions as opposed to with BRAF mutations. So we do not really have that data because we had the single arm trials that do not really tell us what is the normal biology of this patient subgroup. And I think this is a little bit of a problem to understand what will the results be in moving these therapies into first line.
Juan, I think your hand is up.
Yeah, thanks, Anna. And just to follow on from that comment, I think obviously we want to know what the efficacy of using, let's say, a quadruplet combination up front, but we also need to be mindful, what are the aims of our treatment? If we're trying to improve a response rate, for example, by adding a HER2, which would then lead to, tumor reduction or something that's initially inoperable and then go for cure, that seems an appropriate way to do things. If we're dealing with somebody who's got very extensive metastatic disease, maybe sequencing well-tolerated treatments where you're not having to compromise due to overlapping toxicities, for example, might be better. So going for the marathon approach rather than a sprint might be better.
Thank you. I think now we have to close. Thank you so much, Angela, once again for the amazing presentation. Thank you all of you for the fruitful discussion. Once again, this webinar has been recorded and will be on the website, and we will soon share the date for the next webinar. Thank you. And
thank you, everyone. Bye.
Bye. Bye-bye.